Gastroenteritis remains a challenge to diagnose due to a myriad of causes and overlapping syndromes. Molecular panels allow for ease of testing for multiple targets, but are not without their limitations. Here to discuss some of the pros and cons of GI multiplex panels with us today is Dr. Meghan Starolis, one of the authors on the recent review article entitled Viral Pathogen Detection Using Multiplex Gastrointestinal Molecular Panels, the Pros and Cons of Viral Target Inclusion. Dr. Starolis, thank you for joining us today.
Thank you.
Let's start off. Can you tell me a bit about yourself?
Of course. So my name is Dr. Meghan Starolis, and I am currently a council member for the Pan American Society for Clinical Virology, and I serve as chairperson of the Clinical Practice Committee. I am a board certified laboratory director, and my specific professional focus is molecular diagnostics.
Great. And what is the mission of the Clinical Practice Committee for the PASCV?
So our committee has several high-level objectives. Our primary goal is to identify areas in clinical virology where there are diagnostic challenges, and we use our committee of subject matter experts to establish best practice guidelines. Some recent initiatives have centered around measles, arboviruses, and gastrointestinal disease, specifically around the use of syndromic panels for gastrointestinal disease. We also respond on behalf of the society to any public policy concerns related to clinical virology, such as the recent proposal from the FDA to regulate LDTs.
Okay, so let's get into it. Tell us about the recent publication from the Clinical Practice Committee.
Yeah, so our most recent work was a follow-up to a gastrointestinal pathogen guideline paper. Earlier this year, we published a guidance document on the appropriate use of these GI syndromic panels. But we really wanted to follow that up with a manuscript that was more centered around enteric viruses. Specifically, we wanted to discuss the pros and cons of inclusion of viral targets on large syndromic panels in the spirit of diagnostic stewardship. So this particular work was co-authored by myself and two other colleagues on the Clinical Practice Committee. Dr. Eleanor Powell of the University of Cincinnati was the lead author of the manuscript, and Dr. Jane Hada of the Mayo Clinic in Jacksonville also lent her expertise.
Okay, so what are some of the pros of detecting viral targets on GI panels?
There are several arguments for inclusion of viral targets on syndromic panels. From a clinical perspective, it can be very important to get a definitive diagnostic in patients who are immunocompromised, and that is supported by guidelines. Identifying the specific causative agent of disease is also very important for epidemiological reasons to help identify and manage outbreaks. And using a large panel can also speed the time to diagnosis, which can aid in proper isolation for inpatients. It can also help to manage infection control practices and can contribute to antibiotic stewardship if the causative agent is identified as viral.
Those all sound really important. What are some of the arguments then against?
Well, the biggest concern is that inclusion of more targets on a syndromic panel drives up the cost of testing and may not be clinically actionable for specific patients. For otherwise healthy patients, especially those being seen in an ambulatory setting where reimbursement is a big concern, identification of viral causes of gastroenteritis is unlikely to affect patient management. There are no specific treatments for viral causes of diarrhea, and the patient management is really going to be treatment of symptoms. So it could be argued that a smaller panel with bacterial targets or even a stool culture for bacterial pathogens would be sufficient to determine if the patient needed a specific antibiotic. If it was not deemed to be bacterial in origin, really at that point, you're managing the patient's symptoms. There are also some concerns around proper use and interpretation. For example, Most GI panels include a target for C. difficile, which is really not appropriate unless the patient has been hospitalized for several days. Otherwise, it could really just be detecting colonization.
So why not just test everyone for everything and manage the interpretation after the results are determined?
Well, that is a strategy that a lot of people want to use. They want to use these large syndromic panels, but a big consideration when determining if you should use a syndromic panel is how the patient will be billed. For inpatients, this might be less of a concern, and that fast result with a lot of different pathogens may be clinically appropriate. For outpatients, though, who will be using insurance, there are many policies for both private payers and Medicare that limit coverage of syndromic panels. There are specific ICD-10 codes that are required and specific clinical scenarios that they'll reimburse for a syndromic panel. What we don't want is patients being stuck with large bills for a result that may or may not have influenced their clinical care, or laboratories having to absorb the high cost of denied claims for these expensive tests.
Okay, so for those of us with a short attention span, what's kind of the takeaway message? Should viruses be included or excluded?
Yeah, so really the bottom line is that no two patients and clinical scenarios are the same, and a one-size-fits-all approach is not feasible. So just like in all areas of healthcare, we need more flexibility so that we can provide the right test for the right patient at the right time. And what this might look like is the ability to completely mask certain targets so that a more appropriate CPT code could be billed, and also working with our physician colleagues to order the appropriate targets based on what the patient is experiencing.
All right, well, this sounds like a great paper, and I think will be helpful to a lot of us in the lab. Thank you again for joining us today.
Thank you.